Okay, welcome everyone to our next session. I'm gonna grab Maurizio and bring him up on stage to introduce our next speaker.
There we are. Scott, please give me the okay when you want me to start.
Yep, I see you and hear you, so you're good to go.
Perfect. So good morning, everyone. This is Maurizio De Pita speaking, and we are on the third speaker of the session on synaptic physiology. It is my pleasure to welcome here on board Sukbin Lin from the neuroscience program at NYU Shanghai. So Sukbin is a dear friend of mine, and we crossed paths as we were doing both our postdoc training at Nicola Brunel Lab in Chicago. But before then, Sukbin had an excellence pathway that started doing undergrads in Korea. And then she moved with John Rinzel to do the PhD in mathematics. And then she did her first postdoc training with Mark Goldman in UC Davis. And since I believe 2016, 17, wow, God, I'm old. Yeah, she moved to Shanghai, taking on the position of assistant professor there. And we should particularly thank Sukbin to agree at this talk because she's joining us from Seoul, Korea right now. So it's about, by now, midnight. And so she's really making a great effort, and I really appreciate, I believe, on my name and all the audience and the other speakers. Sukbin, without any further ado, the stage is yours, so please go ahead.
Thanks, Mauricio. It's my great pleasure to present my work in this Canadian computational neuroscience spotlight virtually, especially after very inspiring talks and a tutorial by Mauricio, Richard, and Zhang Luigi. So today I will talk about this efficient inference of the synaptic learning rule from in vivo data. And yes, the previous talk is a very nice introduction, and I can be a little bit fast. So the Hebbian learning rule has been well discussed in the previous lecture, which is one of the major doctrine in the neuroscience. And the property of the original Hebs rule can be summarized by the following feature. That is local and unsupervised without the error or the feedback signal from the external system. And it is associative learning rule, capturing the correlative firing of the pre- and post-synaptic neurons. So it can be a substrate for the long-term memory, as shown in the Zhang Luiji's talk. So the broad range of the application of the Hebbian learning rule and its variant form have been investigated previously, and the recent progress of the Hebbian learning rule further highlight the efficiency of capturing the unsupervised feature of the natural learning. And also, it extracts the coarse and invariance representation in the temporal domain, so it can be a substrate to form the context signal or attentional state beyond the traditional memory association. So this Hebbian learning rule can still play an important role in the cognitive functions. And also, the recent work, we evaluate the role of a Hebbian learning rule such that its combination with the supervised learning can demonstrate a good performance and may not be inferior to the entirely supervised learning rule. So given this Hebbian learning rule underlying the various functions in the brain, I'd like to discuss how the specific form of a Hebbian rule, such as the dependence on a pre- and a post-synaptic rates, can be inferred from the data. So previously, the form of the synaptic plasticity rule has been explored in vitro study, where the most of the cells are silent, and the external inputs eliciting some particular patterns of the pre- and post-synaptic activity with a different timing or the frequency only lead to the local changes in the synaptic rates. So we can this in vitro study directly tests the relationship between the activity patterns and synaptic connections, which is called as synaptic plasticity rule. However, if there is an ongoing activity, then synaptic plasticity and the activity of the neurons makes a feedback loop, and the changes of the activity patterns can be utilized as the indirect evidence of the synaptic plasticity occurring in the brain, such as under the perturbation of the inputs or under the perceptual learning task. However, in this task, it is difficult to infer the form of the synaptic plasticity, and then most of the previous work adapted the form of the synaptic plasticity from in vitro data and explained the phenomena, the observed in vivo qualitatively. So here, the question is, what are the synaptic plasticity rules in the cortical circuit we can best infer from the data? So first, in this talk, I would like to consider the experimental data set, the changes of the activity patterns observed during the visual learning. And we have a tremendous ability to recognize whether the object is a novel or a familiar that is called as the recognition memory. So it also that this type of memory occurs more autonomously, like unsupervised manner, through the repetitive exposure to the stimuli. So maybe unsupervised heavy learning in the cortical circuit might be the mechanism of this novelty detection. So neural substrate for the novelty and familiarity detection have been observed in the many brain areas, including the inferior temporal cortex, which is the pinnacle of the ventral visual pathway. And this ITC is thought to be important for the novelty and familiarity detection because the brain region in this area impairs the object recognition memory. So to illustrate the method to infer the form of the plasticity from this in vivo data, observation of these changes of the activity patterns with the learning, we consider the data obtained by the David Scheinberg group. So in this data set, the monkey passively views the screen showing the either novel and familiar stimuli for the short duration. And for each neuron, 125 novel and 125 familiar images were shown and recorded the activity in response to this set of novel and familiar images. And novel images was never shown before the session, and familiar images were intensively trained before the session. So given this observation, this is a summary statistics of the neural activity for the familiar novel and familiar stimuli. So left panel is the time course of the mean response average over all novel response and all novel stimuli and all neurons. And the blue is the response for the familiar stimuli. And right panel is the distribution of the fire rates of one example inferior temporal cortex neuron for 125 novel and 125 familiar stimuli, which is the average over this stimulus presentation period. So in both cases, we can see the decrease of the average response with the familiarity, shown this decrease here, the blue one, and also in the right panel, there is a shift of the activity to the right, to the left. But the selectivity to the stimuli is enhanced with the familiarity. That is, the distribution gets broadened with the learning. And in particular, for the excitatory neuron, not inhibitory neuron, there is an increase in the maximum response, which is shown as this blue, the blue bar to the left, to the right. So in the first talk, I will demonstrate how to derive the form of the synaptic plasticity from the distribution of the fire rates. And then I will shortly mention about its extension to derive the form of the synaptic plasticity with the additional information together with this distribution, the changes in the distribution, but also the changes in the response dynamics. So to illustrate the method, we consider these recurrently connected network models. And in this network, first I discussed the distribution of the fire rates average over this stimulus presentation. So we consider the static model, steady-state model of the recurrent circuit, where the input is the sum of the recurrent input and then external input. And the recurrent input is the weight is sum of the presynaptic weights, and their weight is the synaptic connections. And we consider the one update of the synaptic weights with the visual experience to the particular stimulus. So there is the previous recurrent weight, and the weight is updated when it experiences a particular stimuli, novel stimulus. And we assume this rate-dependent plasticity rule, that is, this delta W depends on the fire rate of the pre and post-synaptic neurons. Then what we have is, so in the experiment, we have the fire rates before learning and after learning, the distribution of the fire rate before and after learning, that is R and R plus delta R. But to infer this delta W from this data set, there are several steps and assumptions we need to make, because this delta W, the change of the synaptic weight, will influence the changes in the will incur the changes in the input. And then also this delta W is shown as the sum over this presynaptic sites. Okay? So to have an inference of the form of the synaptic plasticity rule from this are the changes of the fire rates, we made several assumptions. The first assumption is on the form of the input-output transfer function here to derive this delta I. Okay. So for this, we made two assumptions. One is that the input to the neurons for the novel images follow the Gaussian statistics. Okay, so we have the distribution of fire rate before learning, and then we have a distribution of the input currents before learning. It follows a Gaussian distribution. Then with the assumption of the this monotonic shape of the input-output transfer function, we can map this different quantile of the fire rates and input currents and get the empirical input-output transfer function. Once we have this input-output transfer function, then from these changes of the fire rates, we could infer the changes of the input currents. So we get the distribution of the input currents for the novel stimuli, and a blue one is the distribution of the input currents for the familiar stimuli. Then the delta I, we need to compute the delta I, the input changes, to obtain the delta W. But to get the delta I, the input changes from the distribution of fire rates, we need the additional assumption, that is, for a given neuron, the length of the stimulus among all stimuli does not change, so length of preservation assumption. This assumption is required because we have we couldn't track the fire rates as one stimulus becomes familiar. We have a different set of novel stimuli and a different set of familiar stimuli. So to match the fire rates before and after learning, we assume that the stimuli which elicits the median response before learning will elicit the median response when it becomes familiar. Under this length preservation assumption, due to this limitation of the experiments, now we could calculate the changes of the input currents as a function of the fire rate before learning. And then the final step to get the form of the synaptic plasticity rule is we made the assumption that this learning rule is a separable function of the pre and post-synaptic rates. So under this assumption that the delta W is a separable function of the post and pre-synaptic rates, then this delta I can be, we can represent this post-synaptic dependence as a function of this delta I with some bias term and a normalization, and this normalization term contains the contribution from the presynaptic dependence, and this bias term is containing the input changes due to the changes of the presynaptic fire rates, not only due to the changes of the synaptic rates. So from this separability, we obtain that this delta I, what we have obtained previously, the input changes as a function of the rates, can be a proxy to the postsynaptic dependence of this synaptic plasticity rule. That is, when you had such changes of the distribution of fire rates, that is, the shift of the overall distribution to the left, the depression, there is a decrease in the mean fire rates, while the increase of the maximum fire rates, we could obtain the some postsynaptic dependence showing the depression for the most of the ranges to account for this decrease in the mean fire rates. But also there is a potentiation to explain these maximum fire rates. So using this method, we could apply this method to the individual neurons and obtain this dependence, postsynaptic dependence on the fire rates before learning. And one of the results, as I mentioned, is especially in the excitatory neurons, many of the cells shows the depression for the low fire rates and a potentiation for the high fire rates. And one other interesting feature we also found is that there is the threshold separating the depression and the potentiation, and this threshold is correlated with the overall activity of the neurons, the standard deviation of the individual neuron, the mean or the standard deviation of the fire rates of individual neurons. So such a correlation is suggesting some regulation of the learning rule, the individual synaptic plasticity rule, depending on the activity of this postsynaptic neuron. And such a regulation is a reminiscent of the BCM rule suggested theoretically. So in this theory, there is some regulation of the threshold that if there is too much potentiation over, like if the activity is too high, then there is increase in the threshold so that depression will be dominant. Or like if there is a too low activity, this scale for instance, then the threshold is following this trend so that there will be a more potentiation. So although this is a more temporal, like a description of the regulation of individual neuron, what they have is not the temporal domain, but in the spatial domain, in the different individual neuron, we have such a regulation. Okay. So, so far I have shown that using this distribution, the temporal time average rate, how to derive the form of the synaptic plasticity rule, especially the postsynaptic dependence. So in this one, we consider how the recurrent synaptic plasticity especially shapes the network patterns and stimulus tuning, how it broadens with the learning. But not only these, the changes of the stimulus tuning, there is an important change in the response dynamics observed in this brain area. So here I will briefly mention the extension of this previous work to get the further information about the synaptic plasticity by investigating the changes of the spatial temporal patterns of the network activity. So we will utilize the changes of the response dynamics to infer the form of the synaptic plasticity. So to examine the changes of the response dynamics, here is again the time course of the response, mean response average over neurons and the average across all stimuli. The novel is red and the blue is the familiar. And the right panel is the response for the maximum response to the best stimuli. So as we have shown, like previously in the distribution changes, there is a decrease in the average activity, and then there is an increase in the maximum response. But interestingly, not only these changes consistent with the changes in the distribution of the activity, there is a rebound of the activity in both the mean and then maximum response, which is shown around the 150 milliseconds after the activity onset. So overall, it shows that when it becomes familiar, then the activity becomes more oscillatory. Such an emergence of the damped oscillation with the learning has been observed consistently under the different experimental settings. So the same passive viewing task as in the Scheinberg's data, but with a longer presentation. In this case, previously it was shown for the 200 millisecond, and in this case it was shown for the 600 millisecond. Okay. So same passive viewing task, but with a longer presentation. Now we can see the emergence of the damped oscillation more clearly for the familiar stimuli. And Carl Olsen's group have postulated the role of such a damped oscillation with a familiarity. That is, the changes in the response dynamics can be important for the rapid processing of the familiar stimuli compared to the novel stimuli. So they designed the experiment presenting either two novel images or two familiar images successively. And in this case, for the first novel stimuli, the novel in this case was shown as the blue. And for the first novel stimuli, there is a reduction of the activity and a smaller oscillation in this successive presentation of the novel stimuli. On the other hand, when the novel familiar stimuli were shown, the first response is similar as in the single presentation, so there is a rapid reduction. But from the second stimuli, it shows a stronger oscillatory response. So this suggests that the rapid reduction of the activity for the familiar images make the neurons or the network to be ready for the incoming stimuli and more responsible for the dynamic changes of the stimuli. So here are the three summary of the important changes. That is the reduction in the average response and an increase in the maximum response and a stronger oscillatory response after learning. So given this interesting observation, we first investigate whether the recurrent synaptic plasticity derived from this time average data can reproduce the changes in the response dynamics. Results were not shown here, but it shows that although the decrease in the mean response and the increase in the maximum response could be reproduced as in the time average data, this stronger oscillatory response couldn't be reproduced with the recurrent synaptic plasticity derived from the time average data. So to reproduce such a spatial temporal changes with the learning, we could identify the few important elements of the cellular and cellular and synaptic plasticity properties. And due to the lack of time, I will not go into the detail, but the summary of the main finding is the following. Okay. So one is that the network requires the slow negative feedback, such as adaptation, to account for the suppression of the response to the second novel stimuli, and then also the stronger response for the familiar stimuli, so that the synaptic plasticity in the recurrent connection can enhance the positive feedback within the network and can interact with this slow negative feedback and induce the stronger oscillation after learning. So it requires this slow negative feedback and such as a final adaptation, and there requires an interaction between the synaptic plasticity. And also, the average potentiation is required. So this is unlike the previous finding. So the previous finding is showing that there should be an average depression, although there is a potentiation in the high final rates. But this is required, the average potentiation required to reproduce the in-phase, mean, and the maximum response. And then to account for the decrease in the average response, we require the additional synaptic plasticity. So here, there is the segregated role of the synaptic plasticity in the recurrent connection and the fitful connection, such that the potentiation in the recurrent connection is required for the increased positive feedback, while the average, the fitful connection needs to be decreased to account for the reduction, the sparse representation of the stimuli with the learning. So with these few elements, we could reproduce the the main changes, the qualitative changes of the response dynamics with the learning. But also, we could infer the synaptic plasticity rule, the dependence on the post-synaptic dependence of the fire rates. So, from the distribution of the time course data for the novel stimuli and the familiar stimuli, and as expected, there is the potentiation for the recurrent connection and overall depression in the feedforward connection. And with implementing these two observations, we could reproduce the mean response across all stimuli, the reduction and the rebound in the average response, as well as the maximum increase in the maximum response. Okay, so in this second part, I show that not only using this time average data, if we use these changes in the temporal dynamics together, then we could further identify the role of this slow negative feedback, such as adaptation, and also segregate the role of this feedforward and recurrent connection. As a brief note, such a changes of the response dynamics, the decrease in the mean fire rates with the increased selectivity and the stronger oscillation was not only observed in this inferior temporal cortex during the object recognition, but also in the mice brain, in the mice V1 for the disorientation or the direction selectivity. It was shown that the similar response changes was observed. So to dissect the mechanism, the experimental group in Purdue compared the synaptic strengths in the cerebrocortical connection and the intracortical projection in the naive mice and the experienced mice of this orientation, the drifting gratings, and found that in the experienced mice, that is when the stimuli become familiar, the light evoked response EPSC was significantly decreased in the feedforward connection, while there is an increase in the recurrent connection. So this is suggesting that although there is a different modality and an animal, still maybe there is some general principle of learning this familiar stimuli, like reduction in the feedforward connection and then increase in the recurrent connections. Okay, I hope I have some time to cover this last part. Okay, so
yes, you still have more or less 5 to 10 minutes.
Okay, thank you. All right, so what I have discussed so far is the partial inference of the synaptic plasticity rule, that is either using this time average data or response dynamics changes in the response dynamics. Only the postsynaptic dependence of the synaptic plasticity rule could be inferred from the changes of the single cell activities. In the remaining time, I'd like to shortly discuss the efficient method to infer the complete map, both in vitro or in vivo like conditions. And this work is done by Qixin Yang and Shuri Chen, two amazing research associates in the lab. And to get the idea, we consider the biologically plausible synaptic plasticity obtained in vitro study because we don't know the ground truth of the synaptic plasticity in vivo. So in vitro, the changes of the synaptic connections was measured directly, by varying the relative timing of the pre and post synaptic activity, delta t, or the frequency of the spiking activity of the pre and post together. So as covered in Mauricio's talk, the several models have been proposed to capture such an experimental synaptic plasticity with their parameters fitted to the experimental data. For instance, this is the calcium-based model of the synaptic plasticity whose parameters were fitted to the in vitro data obtained in the layer 5 in the left visual cortex for the different timing or the different frequency of the pre and post-synaptic activity. So to obtain the fire rate dependent synaptic plasticity rule, from this Gaussian-based model. And to mimic the this in vivo-like conditions, we consider the irregular spiking activity, the jittered pre- and post-synaptic firing activity, but with a different rate, frequency, and we obtain the delta W, the changes of the rate. So this color map is showing this the delta W, the dependence of is a dependence on the postsynaptic and presynaptic rates, and this red part is the potentiation, and the bluish color is the depression. So we assume that this mean rate changes as a ground truth of the synaptic plasticity rule, and want to first examine what would be the efficient method to infer the in vitro-like condition. So when you have a direct access of the delta W, then can you infer, this entire map? Of course, if you have this post and presynaptic, the entire manipulation of RA and RJ, then we have the complete map. But there are the two obstacles in getting this learning rule. That is the noisy observation first, and also there is a sparse sampling. So in the experiment as done in the Shostrom in 2001, as we can see, there is a fluctuation in a delta I, here delta W, but also only a few data points could be obtained in vitro conditions. So to reconstruct the underlying synaptic plasticity from this sparse and noisy observation, we want to exploit some property of the learning rule. So previously, when I inferred this postsynaptic dependence, I made the two important assumptions. One is the low rankedness of the synaptic plasticity rule, that is approximating this plasticity rule as a separable function of the pre- and postsynaptic fire rates. And another assumption I mentioned implicitly, this length preservation assumption previously, is kind of assumption of the smoothness of this delta W. And this smoothness is natural because as we change the pre- and postsynaptic fire rate slightly, then we don't expect that delta W changes abruptly. So we exploit the several method to reconstruct the complete map of the from this partial and noisy observation. And we found that the non-parametric method exploiting the smoothness, called as a Gaussian process for regression, was effective. And I will skip some intuition behind this Gaussian process regression. And this is non-parametric, and assuming the smoothness depending on the choice of the corner. And using this GPR, the Gaussian process for regression, we show that the original map could be well recovered with a sparse sampling. For instance, here we have 20 observations, 20 noisy observation, and then the right panel is the recovered delta W. And then this difference between the delta W and original delta W and a recovered one is about the 20% of the norm of the original map. And below is the summary statistics of the performance of the GPR, showing the relative error compared to the this norm of the original delta W, but the different number of the observation and over the different realization of the noise and then sampling. So this arrow is showing the performance, median performance at the 20 observation, and it shows the good recovery in a sparse sampling and the denoising with the increase of number of the observation as expected. So given this success of the recovery of synaptic plasticity in vitro-like condition, we further investigated whether this GPR can be utilized to infer the synaptic plasticity from in vivo-like conditions. So in vivo-like conditions, the direct delta W cannot be obtained, but instead we have, as I have shown before, like changes of the activity, and probably the changes of the input currents might be available. So to illustrate the method in vivo-like a condition, we first simulate the network activity using the biologically plausible model, like the Gaussian-based model. And assuming the fire rate before learning, the Nobel, this red one, and then the Wij and the delta Wij from this learning rule, we could generate the fire rate after learning this familiar stimuli, which is a broadened distribution. And then from these changes of the response dynamics, now we are assuming that we know only the fire rate before learning and after learning and delta I, the input currents, but we don't know the rate before learning and then delta W, the synaptic plasticity rule. And what we want to infer here is from these changes of the activity pattern and the input currents, we want to infer the synaptic plasticity rule under the assumption of the smoothness. Okay. So under the assumption of this, given these changes of the rates and the smoothness, we apply the GPR, and again, that around this 40 observation. In this case, the 40 observation is we are assuming that there are 40 neurons, the postsynaptic neuron activity, but also we know that the associated changes of the presynaptic site together. And then without knowing the Wij, but only the mean of the Wij matters in this inference, then we could infer this delta W. So in this case, in vivo-like condition, what we have is the affine constraint one, not the direct access of the delta W, but under the affine constraint, we could still recover well this delta W from around the 40 number of observation. And to compare the method with the previous one, from the same data set, but without the knowledge of the presynaptic activity, but we have these statistics of distribution of before learning and after learning. Then when you apply the method, the previous method, then we could infer the changes, input changes as a function of the rate, which is a proxy to the postsynaptic dependence. And this postsynaptic dependence, inferred from this data set, actually capture well the original learning rule partially. So if we have the original map using the SVD, then the first singular value corresponding to the singular value, we have the left singular vectors and the right singular vector. And the left singular vector is close to the postsynaptic dependence in this case, so which is shown as a red curve. So this method, the original method, could capture the learning rule well partially. But we didn't have information of the presynaptic site in this case. But when you utilize this simultaneous recording of a pre and a postsynaptic neurons together, then not only the postsynaptic dependence, but also we could infer the entire rule, which captures this the first singular vectors well, the postsynaptic dependence and the presynaptic dependence both well.
So,
yes,
okay.
I'm done.
You're done. Okay.
Yes,
great.
yes. So yeah, in the second part, I show the extension of the previous method utilizing the smoothness of the synaptic plasticity rule, and this one was effective to recover the learning rule from the noisy and sparse observation, and under the affine constraint like in vivo-like condition, but it requires the simultaneous recording of a pre and a postsynaptic activity together to infer the entire rule. Okay. So with that, I would like to thank my collaborator and my lab members. Okay, so I hope that I'm on time.
No, you were doing fine. It's just that we are running a little bit late, and nevertheless, thank you very much for this great question, great talk. And because of the time difference that you are at, and I believe you want to rest, so I will just read to you a couple of questions, and you are allowed to answer them, and then go to sleep and rest accordingly. So the first question is from Helson Pascal, and he's asking if you could explain how the weights are bounded. If you use hard or soft bounds, do you think that could avoid the divergence of weights by playing with model parameters?
So in the W, in the inference of the here I didn't consider any bound on the delta W itself. And then on the weight, yes, that's interesting question actually. So if there is any bound, can it be detected? So for instance, yes, so I have to explain. So in my work, I didn't include the weight dependence. So the delta W is just dependent on the pre and a post synaptic rate, right? So if I include the weight dependence together, then for instance, so there is some soft bound or such that if the weight is approaching to the particular value, so starting point of the W, then it cannot be further increased beyond this bound, right? So then for this inference part, I think it requires the another dimension. For instance, in the second part, the last part of my talk, I have the dependence on the rate, but also it may require the additional consideration on the weight, so that like the inference would be not the two-dimensional map, so it would be like a three-dimensional map in that case. So in that case, I think it would be possible, it would be doable, for instance. But the problem is in vitro-like condition, I think it would be doable to infer from the noisy observation. But then like in vivo-like condition, the problem is we don't know the starting point, the delta, the W itself, right? So we are only assuming we are doing the inference on the mean values over for this inference. So maybe we need additional information, knowing not only the changes of the activity, but knowing the weight before learning also it may be required. But I'm not sure whether the current experimental setting will allow to do that. But that's important point. Yes.
Thank you. And the second question comes instead from James Jonathan. He says, he's asking if you can briefly comment on what is the source of the adaptation in your rate model.
Yes. So in my one, like in the second part, I consider the fire rate adaptation, so the slow negative feedback. So I mean, there is a many cellular mechanisms suggesting this slow adaptation like currents, and I think I explored several different possibility for such a slow negative feedback. So the candidates I considered was this spike rate adaptation, and the other one was short-term depression. It also can give rise to the negative feedback to the system, or some global inhibition, slow global inhibition. So what I have found is why this fire rate adaptation was effective in generating such an oscillation with the synaptic plasticity one was it is some kind of a private to the individual neuron. So if I have this slow negative feedback on the global scale rather than a more individual level, while the synaptic plasticity is generating the clustered neurons, right? Like this enhanced positive feedback in the clustered neurons, then it couldn't generate the oscillation as I expected, like this the short-term depression or slow global inhibition. Those couldn't account for the this generation of the oscillation after learning, but only this fire rate spike adaptation, which is more private for the individual neuron, like the slow hyperpolarization currents, like it can be effective. They could only reproduce the changes of the response dynamics as I observed. Yes.
Okay, thank you. We still have a couple of questions, but unfortunately we are running out of time. So I can forward to you the questions, or maybe I will coordinate with the organizers to find out a way to have a board whereby unanswered questions could be answered by a specific speaker. Sukbin, I still would like to thank you on my name and on the name of the other audience. And thank you for joining us. And without further ado, I move to the next speaker. Thank you very much.
Bye.
Bye.